Okay, I'm going to divide this chapter up into 3 days. The first day is going to be the most brutal, I think, and then we'll go from there. But it seemed to work in the spring. That's why I want to stay with this pattern. Okay, chapter 9, okay, it falls under the subject genetics, okay, and titled Patterns of Inheritance. Now, what we covered in chapter 8, the cell cycle, including meiosis, that information feeds into this chapter, everybody. So when you're studying chapter 8, the cell cycle, including meiosis, meiosis 1 and 2, and the meiotic phase, understand that feeds into this. So genetics is a term that was coined roughly around the early 1900s, between 1905 and 1910. And genetics is defined as the science of heredity. Okay. Now the reason why I'm showing you these figures here, especially this one, is because heredity is defined as the process whereby specific traits are passed on, passed on from parent to an offspring. Today, it's known that it's passed on through your genes. Okay. Now, we're going to go over studies conducted by a monk by the name of Gregor Mendel. But when he performed his what are now referred to genetic studies using the pea plant, it wasn't known at the time that DNA is a compound through which living organisms pass on traits, okay, from one generation to the next. He was just going by what he could see, observable features, sometimes called phenotypes. Okay. So the way I describe it to students is once you define genetics, the science of heredity, then that leads you to heredity. Well, what the heck is that? So then we define heredity, but then we have this word traits. Okay. Well, what do we mean by traits? That talking about the process whereby traits are passed on from parent to offspring through genes. Well, traits are genetically inherited features of an organism. What I mean by features in this class, okay, are observable features. I don't know if I can do this morning. Let's see. Let me go ahead and start warming up. Okay. In other words, you're going to hear me use traits and the term phenotype interchangeably. Oh boy, it's getting worse. Okay, this should be an N here. Okay, I'm going to stop right there. That's pretty bad this morning. Okay, not a good thing. But here, features, what I want y'all to do is make a note that where it says features, you're going to come to know this as a phenotype, okay? Genetically inherited features of an organism. So up here, okay, this is a fruit fly or a gnat, or in scientific terms, Drosophila melanogaster. Eye color. That's the feature or the trait that I'm trying to show you here. Geneticists discovered, and they discovered this around the 1930s, 20s or 30s, that these little guys here that we tend to call gnats or fruit flies, but are called Drosophila melanogasters, that there's a trait for eye color. But that trait for eye color comes in two different features or phenotypes, red eye color and white eye color. Red eye color, okay, is the most common type. So this is what I mean by inherited features, okay, but they're genetically inherited through our genes. So thus here, the purpose of this here, obviously, is to demonstrate how scientists eventually discovered how these traits for eye color, okay, were passed on from parent to offspring through units of DNA called genes. So this is to represent what I mean by traits, genetically inherited features. This is to represent what I mean by genetically. Okay, it's encoded in the DNA what the eye color will be. Now, you're going to hear me use the term alleles frequently instead of genes. So it's important to know this. Please remember this here if you're not familiar with it or if you're not very familiar with it. Genes, okay, are referred to as alleles. I should have put are often referred to as alleles. Now, first of all, what are genes? Genes are the basic units of genetic material, and they're stored in the form of DNA. Some people will say genes are units of DNA, okay, that encode for certain traits. All right. Now, as far as why genes are referred to as alleles, it's down here. Okay. This term alleles is used to imply or to refer to that often for a single trait, there are usually two or more genes that will code for that one trait. Case in point, when we go over Gregor Mendel's pea plant studies. Okay, what he didn't know, but later geneticists discovered, is the reason why pea plants, okay, produce flowers with two different colors, purple and white, is because there are two forms of a gene that code for the pea plant flower color. But one of them codes for purple flower and the other one codes for white. But it's still the same trait, flower color. It's just that one trait, flower color, comes in two different phenotypes, purple and white. Therefore, they started using the term alleles to imply to a person, if I use a term to alleles, now you know I'm telling you, hey, there are at least two or more alternative forms of a gene, okay? And they've arisen by mutation as far as we know, and they'll be found at the same gene loci on a homologous pair of chromosomes. But the catch is this: the two or more alternative forms of the gene code for the same trait, okay, or phenotype. Now, I tend to use phenotype. I tend to place phenotype on the trait, and I'll clear this up when we get into Gregor Mendel's work, okay? So as I said, for pea plants, there's a trait, flower color. That's one trait. But there are two different colors of flower. In other words, two different phenotypes, purple and white. And what they eventually found out is that that's because there are two forms of a gene that both code for flower color for the same trait. So we call them alleles. Now, notice something in this definition here also. The terms gene loci and homologous pairs of chromosomes. Okay. Up here, they're showing you a homologous pair of chromosomes. If you recall, on a human karyotype, if we compare a male versus a female karyotype, at a minimum, okay, this is the question I'm asking y'all. If we compare a human female and a human male karyotype, okay, what are the minimum number of chromosomes that should be homologous in those two karyotypes? Yeah, 22, right? So we're going to focus on these 22, which are known as autosomes. We're going to focus mainly on homologous chromosomes, since we're not just going to be talking human beings now. Obviously, we're going to be talking about plants. But even though plants do have different sexes, we're going to focus on homologous chromosomes for this part having to do with genes called alleles. Now, let me ask y'all this. They're saying this is a homologous pair of chromosomes, meaning they're the same or similar. You can see they're the same length, and where it's pinched in, that's representing the centromere. Sorry, boy, this is bad. Centromere. Why the two different colors, though, everybody? I explained that in chapter 8. Why the two different colors? What do the colors represent? Right. So what we call paternal and maternal. In the previous chapter, when I went over a cell cycle involving meiosis, I said the blue will represent paternal DNA, okay, inherited from the father, and the red will represent maternal DNA, inherited from the mother, okay? So this is a reason for the two different colors, okay? Now, what is known today, what geneticists discovered, is that when they look at a homologous pair of chromosomes, okay, what they found is if they scan the DNA of one of the two homologous chromosomes and they come across an allele or gene, okay, if they can identify the genetic information stored in that allele or gene, all right, then two things have been taken care of, okay? One, they know the gene loci, the location of that gene on that one homologous chromosome out of the two. Based on that one, all they have to do with the other homolog is they don't have to scan it. They just come right across to the same location on the other homolog, and they know, hey, this is where I'll find the other gene that codes for the same trait. Okay, so on a pair of homologous chromosomes, once they identify the gene loci, the location of a gene or an allele on one of the two homologous chromosomes, on the other one they haven't scanned yet, they don't have to scan the whole gene, the whole piece of DNA. They just go exactly to the same location of the gene that was here. They go to that same exact location along this chromosome to the same gene loci, and they'll find an allele that codes for the same trait. So what they found is on a homologous pair of chromosomes, today they know you have multiple gene loci. And where the gene loci are directly or in the same location when comparing the two homologs together, that lets the geneticist know, okay, now I have a pair of alleles that code for the same trait. They're going to be located at the same gene loci on homologous pair of chromosomes. Does that make a little bit of sense, everybody? I know it's early in the morning too. It doesn't help any, but I hope it does. Okay. But if you notice something, okay, allele is the same thing as gene. Okay, but as I said, we use allele to infer there's two or more forms of a gene often. But notice now, here it says dominant, and here it says recessive. Well, as we go over Gregory Mendel's pea plant studies, okay, he's the one credited with this dominant versus recessive. But for him, since it wasn't known at the time, to be honest, DNA even existed, okay, and since he was just going by the trait, the phenotype, he's the one that coined the term dominant phenotype and recessive phenotype. In other words, I have the trait for pea plant flower color. That's the trait flower color. And he discovered that purple was a dominant phenotype and white is a recessive phenotype for that one trait flower color. It comes in two different types. So what happened was when all scientists finally got together on the same page, okay, and I'll give you some dates as we go through this in a bit, and it was finally, everybody finally came to the same conclusion. Okay, it's DNA by which traits are passed on from one generation to the other. That's when scientists started uncovering the genetic information, okay, that coded for different traits or phenotypes. And then they were able to determine, okay, let's say I have two forms of a gene that code for the same trait, okay? But it comes in, since it's two forms, what I'm finding out is it comes in two phenotypes. One's a dominant phenotype and one's a recessive phenotype. What geneticists did is they eventually matched the phenotype with which of the two forms of the gene was responsible for the dominant phenotype that Mendel discovered, and which of the two forms of the allele or gene for that same trait was the recessive phenotype, or tied into the recessive phenotype that Mendel discovered. Then what they did, geneticists, is they say, hey, based on the dominant phenotype, okay, I will use the uppercase letter, okay, derived from the phenotype. So in this case, when you see an uppercase letter, such as a B allele, what they're trying to tell you is this is the dominant allele. So to relate this to Mendel's work that we're going to talk about, since we'll focus mainly on flower color to explain everything, what eventually geneticists did is, since Mendel discovered purple flower color was the dominant phenotype, when geneticists uncovered the one or two alleles that code for flower color, and they managed to figure out which of those two was responsible for the dominant phenotype purple, they simply called that gene that coded for that dominant phenotype purple the uppercase P allele. Okay, so the casing here, the casing they use a letter derived from the dominant phenotype. Okay, the letter they choose is from the dominant phenotype, and they will use an uppercase letter to tie that in to giving the name to the dominant allele. So the dominant allele here is the uppercase B allele. Now, in individuals that have inherited a pair of homologous chromosomes from mom and dad, okay, but on one chromosome, they're carrying a dominant allele for a dominant phenotype, and then on the other chromosome, they're carrying the other form of the gene. This other form of the gene codes for the recessive phenotype, such as white flower color, okay? And what geneticists discovered based on Mendel's work and duplicating his work is that whenever they came across an offspring that had a pair of homologous chromosomes, but on one chromosome they inherited one form of a gene or allele for one trait, and then on the other chromosome they inherited the second form of the gene, but for that same trait, okay? Says on one chromosome it's for purple flower color, and on the other homologous chromosome the allele is the other form, it's for white flower color. What they found is the one that's determined the dominant phenotype is the dominant allele is the one that determines the phenotype, even in the presence of the other form. So in individuals that are described as being heterozygous, that means on a pair of homologous chromosomes, they've inherited two forms of a gene, but they both code for the same trait. But what was discovered is that one of these two forms of the gene, okay, only determines a phenotype. The other one is covered up by this other form. Therefore, the one that is covered up or hidden, okay, that's the recessive phenotype, and the gene that codes for it, the form of the gene that codes for it, is called the recessive allele. In individuals that are heterozygous, what has happened is on a homologous pair of chromosomes, they've inherited a dominant allele represented by an uppercase letter and a recessive allele represented by a lowercase letter. Okay? But as far as their phenotype goes, it's the dominant allele that will determine their phenotype. But both alleles code for the same trait. And again, as we go over Mendel's work, I'll explain it more. Okay? So individuals, okay, that when you look at the same gene loci on a homologous pair of chromosomes, and you see that they've inherited two forms of a gene, but for the same trait, and only one of those genes determines the phenotype of the organism, we say that that individual is heterozygous dominant. And it's in individuals that are heterozygous, okay, that geneticists learned with the help of Mendel in determining which form of the gene is dominant and which form of the gene is recessive. However, if you look over here, they have the word homozygous twice. Well, sometimes what happens is that the homologous pair of chromosomes is that they're identical. In other words, they're not only the same length with the centromeres at the same location, but they're carrying the same gene for the same trait. In other words, either on both homologous chromosomes at the same gene loci, dominant alleles for that same trait are being carried. In other words, two forms of the gene were inherited. It was the same form of the gene that was inherited. Notice these are uppercase letters though, okay? Whenever an offspring on a homologous pair of chromosomes at the same gene loci, whenever an offspring inherits only dominant alleles at the same gene loci on the same homologous pair of chromosomes for the same trait, now what we say is that offspring is not only homozygous, but they're, if I get this to work, but they're homozygous dominant. And we denote it by two uppercase letters representing each of these. But sometimes an offspring can be homozygous. As you can see here, they're not homozygous dominant. Geneticists discovered sometimes when an offspring inherits a pair of homologous chromosomes, one from mom and one from dad, that at the same gene loci, okay, they're carrying genes for the same trait, and these genes are not two different forms. They're the same form for that same trait. In this case, though, they're both recessive. So whenever an offspring inherits a pair of recessive genes or alleles for the same trait, we say they're homozygous recessive. Each little a represents an A at these two gene loci on the same homologous pair of chromosomes, but the loci are at the same location. Okay? And I know it gets complicated, but hopefully, like I said, as I go through Mendel's work, it'll start to clear up some. Okay. So here in the end, what they're showing you is a homologous pair of chromosomes, okay, with three different types of genes, not forms, but three different types of genes that code for three different traits. Thus, they use different lettering to indicate total. We're showing you, okay, three different types of genes that code for three different traits, okay, and how the individual for one of these traits is homozygous dominant. For a different trait, they're homozygous recessive. And for a third different trait, they're heterozygous dominant, okay? Now, Mendel didn't know this, and a lot of scientists didn't know this at the time. Now, there are also two words. As I said, I mentioned phenotype, but there's also genotype. When you hear me use the term genotype or genotypic, what we're talking about is the genetic makeup of an organism. So today, what we do, okay, in genetics, study patterns of inheritance and we go over the basics in relation to Mendel's work. This is Gregor Mendel, the monk, who's considered the father of genetics. Today, we often use what is called a Punnett square. Okay, and the Punnett square can be drawn a little different. Information along the edges has to do with the parents. Information within the boxes has to do with the offspring produced by the parents. Okay, but if you notice here, this Punnett square, and they often do this, is showing you two things. It's not only showing you phenotypes as far as purple flower and white flower. Notice they're showing you different letters. Or I'm sorry, notice they're showing you the letter P, but upper and lowercase. In other words, what they're showing you has to do with the genotype, or what they call the genotypic ratio. And this is what we'll build towards when we go over Gregor Mendel's pea plant studies. Okay. However, for Gregor Mendel, there was nothing known about genotypes and gene ratios because, as I said, to my knowledge, DNA wasn't known to exist. Okay, Mendel published his pea plant studies, okay, in 1866. Obviously, he started the studies before then. I believe it was the late 1850s, but he published his work in 1866. It wasn't until around 1900 that there was a large enough group of scientists that said, wait a minute, this isn't some crazy monk growing tens, if not hundreds of thousands of pea plants and standing there and looking at each of them, you know, like some crazy person. They realized he's onto something, he's ahead of his time. So thankfully, due to that small group, such as Reginald Punnett, who developed the Punnett square, another guy by the name of Bates, who coined the term, I believe, genes or genetics, these guys noticed he was onto something. Okay, and then over time, and they were the ones that said, okay, even though DNA was not known to be the compound through which traits were passed on from parent to offspring by the early 1900s, they knew Mendel was still onto something. Okay, so they coined the term genetics, like I said, the science of heredity, how traits are passed on to offspring, and they knew Mendel based his studies on just the phenotype. Okay, well, the trait is a physical makeup of an organism. So when you hear me use the term trait, I'm talking about the physical features of an organism, but we often use the term phenotype. The observable, that's what we mean. Okay. So here in the Punnett square, where they show you purple flowers and white flowers, now what they're focusing on is the phenotype. And eventually, as we go over use these Punnett squares to explain Mendel's work, we'll also draw conclusions from these Punnett squares in regards to phenotypic ratios. But notice they're talking about the color. In other words, what's observable. So there's genotype versus phenotype. Mendel's work, Gregor Mendel's work, was based strictly on phenotype. But because of what is known today, okay, you're often given the impression that he knew about genes and DNA and all that, and it wasn't known then. Any questions right now? I know, like I said, it's not usually easy material to talk about, and it's even harder to try to explain. Okay, hopefully y'all aren't asleep. All right, so listen carefully. Gregor Mendel, the father of genetics, okay, he's the one that uncovered patterns of inheritance, how traits are passed on from parent to offspring. And he did this by studying pea plant phenotypes. Okay. Now, when he studied pea plant phenotypes, he focused on traits, observable features. There were a total of 7 traits that he studied in regards to how they were passed on from parent to offspring. Okay, so there were a total of 7 traits. But what he discovered is that each trait came in 2 forms. Oops, what happened? Came in 2 forms, a dominant and a recessive. Let me use this little line here to represent and. Oops. Almost hit the wrong button and lost y'all. All right. So, again, Gregor Mendel, the father of genetics, he's the one credited with uncovering how traits are passed on from parents to offspring, okay, what we call patterns of inheritance. His studies were based on the pea plants and their phenotypes. And what he studied, what he focused on, were 7 observable features: flower color, flower position, seed color, seed shape, pod color, and stem length. And what he discovered is for each of the traits, they came in 2 physical types or two physical observable features. Okay. These traits came in a dominant phenotype, such as for flower color. Purple is the dominant phenotype for this trait, flower color. But these traits also came in a recessive phenotype, such as for flower color, which is the one trait. It doesn't just come in a dominant purple, but sometimes the flowers appear as a recessive white for this one trait, flower color. Okay, so we have one phenotype, but that is, again, that one phenotype can come in two forms. We have one trait, but that one trait can come in two phenotypes, the dominant phenotype and a recessive phenotype. In either case, this is all observable. Please listen to me carefully, okay? So you don't give yourself extra work. There are 7 traits Mendel studied. You are only responsible for 3. So listen carefully. Going into the third exam, okay, any questions I ask you about Gregor Mendel's pea plant studies and the patterns of inheritance, these are the 3 traits I want you to focus on. Seed shape, seed color, and flower color. So on this PowerPoint, I put arrows next to the ones you need to focus on. Okay. Now, when Gregor Mendel performed his pea plant studies to see how traits are passed on from parent to offspring, thus creating a pattern of inheritance, okay, he conducted 2 types of experiments. One's called a monohybrid cross, and one's called a dihybrid cross. Okay. For the experiments he conducted that are called monohybrid crosses, the mono meant that he focused on only one of these traits at a time, focused on only one trait at a time to determine the pattern of inheritance, how it's passed on from parent to offspring, and then from offspring to the next generation of offspring. After he did that for each of these traits individually, after he performed monohybrid crosses, focusing on just one trait at a time, okay, then he stepped it up. He increased the complexity of his experiments by performing dihybrid crosses. And for dihybrid crosses, whereas mono means 1, di means 2. For the dihybrid crosses, he focused on how a pair of different traits, such as seed color and seed shape, how are they passed on at the same time, from parent to offspring, and then from offspring to the next generation of offspring. Okay, so total he had 2 different experiments. Mono means 1, di means 2. But the reason why he called them monohybrid and dihybrid, the hybrid, had to do with he always started the experiment with 2 separate pea plants. Why? Because pea plants are capable of both asexual reproduction and sexual reproduction, which I'll explain in a second. Okay. Now, so again, these are the 3 traits I want you to focus on. And when we go over his first set of experiments called monohybrid crosses, we will focus on this trait. Okay. And once you come to understand the pattern of inheritance for flower color, how purple and white is passed on, then it's what I call a pull and pluck. Any information I give you for the dominant phenotype, or the dominant phenotype for flower color purple, you would pull that out and insert yellow, because for seed color, the dominant phenotype is yellow. Any information I give you for flower color that it has to do with white, you would just pull that out, and for seed color, you would insert green, because the pattern of inheritance in a monohybrid cross is found to be the same for all of these. Okay, so another thing you'll need to know going into the test is make sure you know for this one trait flower color, it comes in 2 phenotypes, a dominant phenotype purple and a recessive phenotype white. For this other trait, seed color, it also comes in 2 phenotypes, a dominant phenotype yellow and a recessive phenotype green. And then for another different trait, a third different trait, seed shape, it also comes in 2 phenotypes. Okay, the shape of the seed could be a dominant round, or the shape of the seed could be a recessive wrinkled. Okay, so that's the first thing you want to know as you're going through this before you proceed any further, okay? Okay. Now listen carefully. I haven't been able to find it find out or not if Mendel just got lucky from the beginning of his studies and just happened to pick the right organism, a pea plant, or if he actually looked at different organisms or at least different types of plants and decided to focus on the pea plant because he felt like he could interpret the results much easier. Okay, because the pea plant worked out real nice. As science advanced and technology advanced, geneticists today know that Mendel's the conclusions Mendel drew from his experiments, they are true, but they're true for the pea plant. They're not true for all organisms. Sometimes it's much more complex, such as Drosophila melanogaster, that fruit fly. Sometimes when they study a trait in that fruit fly, it pretty much follows a pattern of inheritance in pea plants, but there are other traits in the fruit fly that do not follow patterns of inheritance of a pea plant complex. So this made even a bigger bonus for scientists when they realized Mendel was onto something that now we consider genetics, is that his experiments, it's as simple as we can get for the field of genetics, okay? But another thing about pea plants is plants in general, including pea plants, okay, are capable of two types of reproduction, okay? Plants can produce offspring either asexually, okay, asexual reproduction. In other words, a plant can self-fertilize itself and produce its own offspring. So asexual reproduction requires only one parent that can produce both the egg and the sperm, and therefore self-fertilize itself and produce its own offspring. But these same plants can also sexually reproduce. Sexual reproduction requires two separate parents, one female and one male. The female parent plant produces the egg, the male parent plant produces the sperm, and that sperm will fertilize the egg, producing offspring. Sexual reproduction for plants is also called cross-fertilization. Okay? So asexual reproduction is also known as self-fertilization, and sexual reproduction for plants is known as cross-fertilization. And cross-fertilization requires two parents. Why is this possible? Well, pea plants are flowering plants, everybody. Okay, and the majority of known living plants today are flowering plants. But what a lot of people don't know is that a flower is a reproductive structure, or it contains reproductive organs. I should have put contains. Okay. In a flower, you have these little thin structures with these little expanded tips that we call the stamen. The stamen is associated with the male part, okay, of this flowering plant. It's where sperm are produced. But in this same flower, you have this bowling pin looking thing, this bowling pin looking green thing here, okay? And within this bowling pin looking thing called the carpel, okay, this is a female part of this flowering plant. It's where the egg is produced. This is what allows for pea plants to self-fertilize themselves. But Mendel realized that he could manipulate the process. He realized that, hey, I can take a pair of scissors and cut all these stamen off, okay, thus removing the potential for sperm produced in the stamen to fertilize eggs produced in the carpel. So he would physically remove from some of the pea plants the part of the pea plant responsible for the production of sperm, okay? But he would only do this when he wanted to perform cross-fertilization. And I said cross-fertilization means sexual reproduction. So what they're showing you here is Mendel was getting the pea plants ready to sexually reproduce through a process called cross-fertilization. With this paintbrush, he would take some of the pollen off of the stamen, which contains the sperm, come over here to the carpel, okay, and fertilize the eggs within the carpel so that they would produce the next generation of offspring. Now, the plants he started with, we call them the P generation. The P stands for parents, okay? So whenever Mendel started a monohybrid or a dihybrid cross, he always started with cross fertilization. Okay. That first generation that he cross fertilized, this was our parent generation, and it involved two separate parents. One served as the male, the other served as a female. Doesn't matter if it's monohybrid or dihybrid. He always started with a P generation that had two separate parents so that they could sexually reproduce, and he would do a cross fertilization. This P generation would always produce a group of offspring called the F1 generation. The F comes from a word, I'm not going to go over it, okay, because it's not very popular to go over it. I don't know who coined it, but it just adds to confusion. But we call this generation the F1 generation. So for this F1 generation, the genetic information and thus the phenotypes or the traits this F1 generation inherits, it's coming from a combination of these two parents. Okay, so what they're showing you here represents specifically a monohybrid cross. Okay, what he's focused on in this cross has to do with flower color, purple versus white. So if you look at the pea plants here, notice they're trying to show you when Gregor Mendel performed monohybrid crosses, focusing on one trait, which had to do with flower color. What he discovered is when he cross-fertilized two separate pea plants, one that produced purple flowers and one that produced white flowers, it always produced an F1 generation of offspring that only grew purple flowers. Well, this is a catch, okay? But initially, when scientists looked at Gregor Mendel's work, they asked, okay, but what about these two individual plants? Before we use them for the monohybrid cross, how do we know they're what we call pure breeds today, or what they called back then true breeds? Okay. Mendel made sure to explain in his publication that whenever he was going to perform a monohybrid cross or a dihybrid cross, the initial plants he used in the P generation, he made sure to clarify that these plants were true breeds. Today we don't say true, we tend to say pure. But for Mendel's studies, he said pure. So listen carefully. What Mendel did before he ever started the monohybrid cross, he would take, say, just this one parent here that produced purple flowers. Okay, and imagine we get rid of all this other stuff here, and we're only looking at this flower. What he would do then is he would take that pea plant that produced purple flowers, and he would allow it to keep self-fertilizing itself to produce a next generation of offspring. Then he would take those offspring and let them produce another generation. Until eventually he found that he had plants that no matter how many times they self-fertilized to produce the next generation of offspring, the flowers were always purple. Then he knew he had a true breed that could only pass on purple flower colors to the next generation. Then what he did is he focused on pea plants to produce white flowers. Now imagine we get rid of all this now, get rid of everything here, and you imagine we're only looking at this, a pea plant that produces white flowers. What he did then is he would allow these pea plants to produce white flowers to self-fertilize to produce the next generation of offspring. Then he would allow those offspring to self-fertilize to produce the next generation until he ended up with a true breed. A pea plant that whenever it self-fertilized, no matter how many times it self-fertilized, it only produced pea plants that grew white flowers. Once he had these two true breeds, then he took each separate true breed and performed his monohybrid cross, focusing on one trait, flower color. The hybrid has to do with one of these true breeds, okay, before the experiment started, would only produce through self-fertilization pea plants with purple flowers. The other true breed through self-fertilization before this experiment would only produce offspring with white flowers. Once he had those two true breeds, then he was ready to begin his monohybrid crosses. He would take these two separate true breeds and they would represent his P generation. But now he would do cross-fertilization. And what he noticed is that despite the fact by itself, this plant here, when it's self-fertilized, would produce offspring with white flowers, demonstrating this trait for white flower color could be passed on to the offspring. When he cross-fertilized these two, he ended up with results that were very different. Now he discovered, wait a minute, now when I cross-fertilize these two true breeds, all of a sudden the trait for white flower color has disappeared in my offspring, my F1 generation. And he did this tens of thousands of times, and the results kept coming out the same. From these two separate true breed parents, one that produces purple flower colors and one that produces white flower colors, he would always lose the phenotype for the one that grew white flowers. Only this parent's phenotype would appear, okay, in the F1 generation. So what Mendel wanted to know, okay, I'm trying to write the word hidden. What Mendel wanted to know then is if the trait for white flower color was hidden or lost. For this F1 generation. Now again, remember, ignoring all of this, if this parent here was allowed to self-fertilize, it would produce offspring that grew white flowers. So Mendel knew this trait for white flower color could be passed on to the next generation through self-fertilization. But when cross-fertilizing two separate true breeds, this trait for white flower color vanished. So he wanted to know, okay, is this trait for white flower color hidden in these plants and I just can't see it because it's not observable? Or is it lost? Is it gone forever? Therefore, for Mendel's monohybrid crosses, mono meaning he's focusing on one trait, in this case it's flower color. Hybrid meaning that for his P generation, he used two separate true breeds, okay? And the two separate true breeds were cross-fertilized, okay? From that, no matter how many times he reproduced this experiment, he always ended up with an F1 generation of offspring that only produced purple flowers like one of the two parents. So what happened to that white flower color trait? So therefore, Mendel said, okay, for my monohybrid crosses, I need to take it a step further. Okay? I need to take this F1 generation of offspring and I need to isolate them from each other. Okay? And I want them to asexually reproduce through self-fertilization. So I'm just going to put self-fert because obviously I can barely write with this thing. Okay? So again, from the P generation to the F1 generation, okay, P generation produces the F1 generation offspring through cross-fertilization. Two separate true breeds that are cross-fertilized producing the F1 generation of offspring. But because Mendel wanted to know, since all these F1 generation offspring plants only grew purple flowers, he wanted to know, what happened to this other trait? I mean, this parent contributed to producing this generation of offspring. What happened to white flower color? Is it hidden in these F1 generation offspring, or is it lost forever? And from now on, only purple flowers will appear in any offspring. So what he did to answer his question, is this trait for white flower color hidden or lost? He separated each of the F1 generation offspring from each other and allowed them to self-fertilize themselves and produce the next generation of offspring called the F2 generation. Okay. And when he did this, okay, he did this for two reasons, obviously. One, I want to know is the trait hidden or lost in these guys. But the reason for the self-fertilization is he didn't want to introduce, okay, any more traits. In other words, I only want to know, is that trait for white flower cutter hidden or lost in each of these offspring, one at a time. I only want to investigate what's inside of them, so I don't want to introduce any separate other plants. I just want to see what they have. The best way to do that is let them self-fertilize. Whatever's in this one plant, when it fertilizes itself, it can only pass on what's in it, hidden or not. So when self-fertilization occurred for each F1 offspring, it produced the F2 generation. And he got his answer. From this parent plant that produced white flower colors, okay, that was one of two parents producing the F1 generation offspring. What Mendel discovered through self-fertilization of each individual offspring producing the F2 generation is that from this P generation plant that produced white flower colors, that color for white flower, that trait, it wasn't lost, it was hidden. Okay? So what he discovers is even though none of the offspring ever produced white flower colors, he discovered that these plants were carrying the information for this trait for white flower color, but it was hidden. And how did he prove it was hidden? Well, it's when he took these F1 plants and allowed each one to self-fertilize itself, producing the F2 generation. What ended up happening, he produced thousands, he allowed each individual F1 offspring to self-fertilize and produce thousands of plants. And on average, what Mendel discovered is that for every 4 F2 offspring produced by one F1, okay, on average, 3 out of 4 would produce purple flowers. Now he's counting thousands of plants, but on average, 3 out of 4 would produce purple flowers in the SF2. However, 1 out of 4 would produce white flowers. So what did that tell him? Even though this F1 generation, none of them ever produced white flower colors, that doesn't mean the information for white flower colors wasn't passed on from this one of two parents to its offspring. It was just hidden. Now, he didn't know how it was hidden, but he knew it was hidden. And where was the proof? When he allowed one of these plants at a time to self-fertilize and produce the next F2 generation. On average, 1 out of 4 would produce white flowers, which you never saw in the F1, but now every 1 out of 4 F2, you did see it reappear. Okay? And this was key for geneticists, because that told geneticists later by the 1900s, okay, Mendel's onto something. We don't see the trait for white flower color where it's hidden. Well, a lot of students will say, well, what's the big deal? It's plant flower color. Well, it can have a negative spin to it. Cystic fibrosis is what is referred to as a recessive genetic disorder. You could be carrying the genetic information from it and not even know it because it's hidden. Sickle cell disease, okay? The person that has sickle cell disease has to inherit 2 mutated recessive alleles. Now, they can have some negative impacts on their phenotype if they inherit just one, but the thing is, it could be hidden in them. So this is where it came into play in the future, okay, that it was important to know sometimes, okay, information for a trait, even though it doesn't appear in the offspring, that doesn't mean the offspring's not carrying it. It might be hidden in the offspring, and you don't even know it. So this is what it proves. So listen carefully. For Mendel's monohybrid crosses, okay, whenever he carried out his monohybrid crosses to the point that he produced an F2 generation, that F2 generation, okay, phenotypically speaking, that F2 generation always resulted in a ratio of 3 to 1, okay? And not any ratio, it's a phenotypic ratio. Let's see if I can write this out, okay? It didn't matter if the monohybrid cross focused on the one trait for flower color, or if the monohybrid cross focused on the one trait for seed color, or if the monohybrid cross focused on the one trait for seed shape. He always got the same pattern of inheritance, okay? One of the two traits was always dominant and only appeared in the F1 generation, And then, no matter which of the three traits he was looking at, flower color, seed shape, when he allowed that F1 generation to self-fertilize, producing the F2 generation, he always ended up with a phenotypic ratio of 3 to 1. 3 dominant to 1 recessive for every 4 plants counted. So instead of saying 3 out of 4 or 1 out of 4, we say the phenotypic ratio is 3 to 1. Okay, make sure you make a note. The 3 is in reference to the dominant phenotype, and the 1 is in reference to the recessive. Okay, and it's just pull and pluck. Okay, so if we go back here, look at seed color. There's a trait for seed color that comes in both. Come on. Sorry, everybody. I'm moving the cursor around and it's not writing sometimes. Okay, for seed color, that's the second trait. Dominant phenotype is yellow color. Recessive phenotype is green color. So all you would do is wherever it says purple flowers, you would put yellow seeds and draw a yellow seed. Wherever it says white flowers, you would put green seeds and draw a green seed, and then just pull and plug. F1 generation would have had produced all yellow seeds. Self-fertilization of the pea plants, the F2 generation, would have resulted in a 3 to 1 phenotypic ratio. On average, 3 plants would have produced yellow seeds to 1 plant producing green seeds. Does that make sense what I mean by pull and plug, everybody? It's an athletic term, to be honest, football term. Are y'all still with me? Or did I make y'all run away? Okay. All right. Okay, if it feels like I'm being abusive right now, remember, I get paid for it. That's my incentive. It's my job, okay? So sorry. Got to pay the bills. All right, so listen carefully. Geneticists came along much, much later, okay? I don't know the exact timeline, but I assume it has to be after the 1950s because Mendel published his pea plant studies by 1866. A compound was discovered in the 1860s, around that time, but the scientist didn't know what he was looking at. It wasn't until the late 1870s that a chemist came along and said, okay, serious compound, I've been able to uncover a few characteristics, such as it has these molecules that today we know as bases, AGCT, but still wasn't sure what it was. By the early 1900s, the majority of the scientific community was saying, hey, look, man, what Mendel discovered, these traits and how they're passed on from parent to offspring and these patterns of inheritance, it's got to be due to proteins. It's got to be proteins that they're passing on to their offspring, okay, that's responsible for these patterns of inheritance in regards to traits. But there was this other group of scientists saying, no, we don't think it is. We think the proteins are directly or indirectly responsible for the physical features, the phenotypes. But we believe that the way these proteins come to exist in the offspring is through DNA, genetic information stored in DNA, passed on to the offspring, and that genetic information tells the cell how to make the proteins that will be responsible for the phenotype. But scientists said, no, because so far by the early 1900s, many scientists said, no, there's no way. Look at the diversity in life. We have living organisms that are prokaryotic, and we have living organisms that are eukaryotic. We have single-celled, unicellular living organisms and very complex multicellular living organisms. There's no way it could be DNA. When we look at DNA, the only real difference is that there's 4 bases. Look at proteins. They're so diverse. They perform so many different functions. So a large part of the scientific community by, say, the 1920s and 30s were saying, no way. As a matter of fact, there was a scientist by the last name of Morgan, okay, who was also given a lot of credit for contributing to the field of genetics, and he won the Nobel Peace Prize, but he was adamant it's not DNA. Yet he's considered one of the main contributors, arguably second only to Mendel, but he said it wasn't. It wasn't until the 1950s that it was conclusively proven, and I'll give you a couple of examples after the third test of a couple of 2 groups, that it was conclusively proven. Look, everybody, as simple as DNA is structurally, it is responsible for all the diversity in life. It is the compound used to pass on traits from one generation to the other. So once that was conclusively proven to where the scientific community could no longer argue the point, okay, that's when geneticists could absolutely go back, and also technology improved. Okay, they could do what we call DNA sequencing to uncover the specifics of genetic information. Once all this was identified, geneticists went back and they duplicated Mendel's work. We're going to create true breeds. Then we're going to take these true breeds, such as a true breed that when it self-fertilizes only produces purple flowers, and a true breed that when it self-fertilizes only produces white flowers. Then we're going to sequence its genetic information, okay? And we're going to uncover the genetic makeup or the alleles. Well, what they found is that the true breeds Mendel used, such as the one that only produced purple flowers, well, the reason why when this true breed self-fertilized and that always resulted in offspring that only produced purple flowers is because it contained a homologous pair of chromosomes that were both carrying a dominant allele for purple flower color. So in other words, the true breed was homozygous dominant for purple. Therefore, through cell cycle involving meiosis, it did not matter how many gametes this parent produced. That gamete was always going to be carrying a chromosome with the dominant allele. So listen carefully here. They're showing you a pair of peas, but you've got to imagine that you're looking at this. Okay? So when I show you a pair of letters, you've got to imagine, okay, that pair of letters, which represents alleles, that they're part of larger structures, God darn it, part of larger structures that we call homologous chromosomes. And this is where a lot of confusion comes from. The students start seeing just the letters, like down here, and they don't realize, yeah, but you've got to imagine that these are units of DNA that are part of larger structures called homologous chromosomes, okay? And what they found for the other true breed is that it had a pair of alleles on a pair of homologous chromosomes for the same trait, flower color, except these alleles are coded for genetic information that's now referred to as recessive. So what they found is the other true breed used for the P generation in his monohybrid crosses, it wasn't homozygous dominant, it was homozygous recessive for white flower color. But since it was homozygous recessive, since on its homologous pair of chromosomes, it was only carrying the recessive allele, okay, it didn't matter how many gametes this true breed produced. The true breed, okay, would produce gametes that are always carrying a chromosome with the recessive allele. So when Mendel cross-fertilized this pea plant that was homozygous dominant for purple flower color with this pea plant that was a true breed but homozygous recessive for white flower color, when this gamete fertilized this gamete, okay, we ended up with an F1 generation of hybrids, thus the term hybrid here, okay. We ended up with an F1 generation of offspring or hybrids that genetically speaking were all heterozygous dominant. Each of these parents passed on a homologous chromosome. Each parent passed on a chromosome, okay, that were a homologous pair of chromosomes. And these homologous pair of chromosomes at the same gene loci were carrying alleles for the same trait flower color. But from this parent on its homolog, it was carrying an allele for a dominant flower color purple. And for this parent that passed on the other homolog at the same gene loci, it was carrying genetic information for flower color, but it was for white. And if you recall, the way geneticists determine which piece of genetic information is the dominant allele and which gene is the recessive allele is when they look at individuals that are heterozygous. And what they found is that the genetic information for flower color was the dominant genetic information. It determined the flower color. This other form of the gene for flower color, it was recessive. It was covered up by this. So what they found is the reason why the F1 generation plants all produce purple flowers was because they were heterozygous dominant. However, that also explained how the trait for white flower color could be hidden. Remember what I said, the first day will be painful, then hopefully won't be as bad the next days. So what they discovered, geneticists, is the reason why the F1 generation only produced purple flowers was because they were all heterozygous dominant. And this dominant allele coded for the dominant phenotype, purple flower color. Okay? But that didn't mean that the information for white flower color was lost. Okay? It was hidden, but it was hidden in a recessive allele. Okay? Which they now signify as lowercase. So notice, the alleles for flower color, for pea plants, okay, since purple flower color is the dominant type, then we gene the uppercase P allele to represent the phenotype, the dominant phenotype, purple flower color. And we use the lowercase letter from this dominant phenotype, lowercase p, to represent the recessive allele, lowercase p allele. Except in these F1 plants, when they self-fertilized, okay, in the stamen where gametes called sperm are produced, okay, these sperm called gametes would either contain the chromosome with the dominant allele, but half of these sperm that were produced in the stamen of the pea plant would contain the chromosome with the recessive allele for flower. So 50% of the sperm were carrying the chromosome with the dominant allele, 50 were carrying the recessive allele. But in that same F1 plant that was going to self-fertilize itself, in the carpel, gametes called eggs were being produced. And 50% chance they were carrying a chromosome with the dominant allele for purple flower, 50% chance they were carrying a recessive allele for recessive color, white flower. So what happened was Reginald Punnett came along and said, look, let's try to simplify this if we can. Let's take the genetic and the phenotypic information. So notice it says phenotypic and genotypic. Okay, and use a Punnett square and apply the Punnett square to explain the F2 generation. Who produces the F2 generation? The F1. How is the F2 generation produced? Well, I said before, through self-fertilization. Why self-fertilization? Because Mendel wanted to know what happened to the trait for white flower color in this F1 generation. Is it hidden or is it lost? So Reginald Punnett says, okay, let's do it this way. For one F1 offspring, okay, in its stamen, it can produce sperm. These sperm, okay, are either going to be carrying a homologous chromosome that has one homolog that's carrying the dominant allele, or the other homologous chromosome that's carrying the recessive allele, 50-50%. And in the carpel of that one F1 plant where the eggs are being produced, okay, down here, 50% of those eggs are going to be carrying a chromosome that's carrying the dominant allele. 50% of the eggs are going to be carrying the other homolog with the recessive allele. So then Reginald Punnett says, let's line them up this way. I have a habit of drawing the line out a little further sometimes. So what's drawn outside of the enclosed boxes, here for the sperm and here for the eggs, that's information about the F1 parent that's going to self-fertilize. And then what Mendel says is, let's do it this way. Let's here where the sperm is. And let's say this sperm fertilizes this egg. Now what happens? The offspring will inherit a pair of homologous chromosomes, okay? One maternal, one paternal. But if we look at the alleles for flower color, this offspring is going to be homozygous dominant, meaning they're going to produce purple flowers. He says, but what happens if this sperm doesn't fertilize this egg, but instead it fertilizes this egg? Now what happens is we'll end up with an offspring that is heterozygous dominant. It has a pair of homologous chromosomes, one carrying a dominant allele, one carrying recessive. However, the phenotype is still going to be what? Purple. Because the genetic information carried in the dominant allele, okay, hides or masks the information covered in the other allele. but they both code for flower color, but we only see the results from one in the phenotype. Then Reginald Punnett says, yeah, but it's just as possible, by chance, that it's this other half, this other group of sperms carrying the recessive allele that are responsible for fertilization. So let's say this other 50% of sperm, one of them carrying the recessive allele, fertilizes this 50% of eggs carrying the dominant allele. He goes, well, we end up still with a plant that is heterozygous dominant, genotypically speaking. Phenotypically speaking, it's going to produce purple flowers. He says, yeah, but there's just as much chance that this sperm carrying this recessive allele fertilizes this egg carrying the recessive allele. And Reginald Punnett says, and this explains what happened to the trait in the F1 generation, why it's hidden. There are two different alleles that code for flower color. One of the two alleles codes for recessive flower color white. And the only way we're going to see this recessive phenotype is if the offspring is homozygous recessive. Okay? This other allele, the dominant allele, its genetic information, okay, it dominates. It determines what the phenotype is going to be, even in the presence of the other alternative form of the allele recessive. So Reginald Punnett says this Punnett square demonstrates that in order to see this other type of flower color, the recessive phenotype white, the offspring is going to have to be homozygous recessive. And that's what the parent was here, and that's what the grandchild is here. So then from there, what we do, okay, is we extract the information from this Punnett square to draw a conclusion. First conclusion we want to draw has to do with phenotypic ratio. This explains the 3 to 1 using Reginald Punnett square. On average, despite the thousands of F2 generation plants Mendel produced, on average, 3 would produce purple flowers and 1 out of 4 would produce white. Well, Reginald Punnett's Punnett square demonstrates that. On average, 3 have the phenotype purple flower color, which is dominant, and 1 out of 4 has white flower color. So Reginald Punnett said, we don't need to do this anymore. We don't need to spend all this time producing all these plants. We have enough already. We have enough, how do you say, there have been enough experiments conducted with the living organisms that now we can just put it down on paper to explain things. But from this Punnett square, we can also extract second, genotypic information related to the genotypes. Okay. Even though the phenotypic ratio is 3 to 1, 3 purple to 1 white, okay, at the level of the gene, the genotypic ratio is different. Okay, of the 3 that produce purple flowers, only 1 at the level of the gene is homozygous dominant. The other 2 that produce purple flowers, they're actually heterozygous dominant, meaning that they have the potential to produce offspring that won't produce purple flowers like them. They might just by chance produce white flowers. Whereas this 1 of 3 that produces purple flowers, it can only pass on a dominant allele. Any offspring it produces through self-fertilization, asexual reproduction, or cross-fertilization, sexual reproduction, is going to have a purple flower. It can only pass on a dominant allele, like this grandparent. However, the 1 out of 4, okay, which produces white, its genotype is homozygous recessive. For the phenotypic ratios, there's only 2 phenotypes, purple and white. Purple is dominant, and we get a 3 to 1 for the F2 generation. However, the genotype, there are 3 different genotypes that form the genotypic ratio, a 1 to 2 to 1. 1 homozygous dominant, 2 heterozygous dominant, and 1 homozygous recessive. So it's more complex here. So from Mendel's monohybrid crosses, okay, he drew a couple of conclusions. His first conclusion is that there are dominant phenotypes and recessive phenotypes. Today, geneticists have added in information, and they say from Mendel's work that they've duplicated and knowing that traits are passed on through units of DNA called genes, now they credit Mendel with uncovering our dominant alleles that code for dominant phenotypes, and there are recessive alleles that code for recessive phenotypes. So Mendel gets the credit not just at the phenotypic level for dominant and recessive phenotypes, Mendel also gets credit at the genotypic level, or at the level of the genotype, dominant and recessive genotypes. And then Mendel himself, okay, drew a conclusion from his monohybrid crosses. Okay, and he simply called this his first law, and the name of this first law is the law of segregation. And it states, gametes carry only one allele for each inherited trait because the allelic pairs are segregated from each other during meiosis 1. Okay, what he's saying is, if you look here, we have what is called a tetrad, which is made up of a homologous pair. We have a second tetrad, which is made up of a homologous pair. Okay, talked about in chapter 8 during cell cycle involvement meiosis, specifically meiosis 1. Okay, the purpose of meiosis 1 is to take a diploid nuclei, transform it into two separate haploid nuclei. What do we mean by that? We want to segregate or separate these homologous pairs. I want to take one of these two homologs and package it in this haploid cell. I want to take its other homolog and package it in this haploid cell. Okay? Therefore, if there are alleles, God, the color's not coming out here. Okay? Therefore, if we were to look at just this homologous pair, okay, and we were to say at the same gene loci, okay, we find a pair of alleles for the same trait, but let's say on the paternal DNA, okay, I have a dominant allele, say an uppercase P, and let's say on its homolog here, I have the recessive allele. I'm going to use a lowercase p, and I like to put a little tail on the back. Okay? So I have a homologous pair, but on this one here, the paternal, it's carrying the dominant allele for a trait. And on the maternal, it's carrying a recessive allele, but for the same trait. But the thing is, during meiosis 1, this homologous pair is segregated. So now what happens is in this haploid cell, we have one of the two homologs that's carrying the dominant allele, and the maternal one is no longer paired together with its homologous paternal DNA. It's been segregated into a separate cell. So this is what we mean. Gametes carry only one allele for each inherited trait because the allelic pairs, which I'm showing you here, this is the allelic pairs, represented here, okay, are segregated from each other. That's what's represented here during meiosis 1. So for your homologous chromosomes, those first 22 pairs, okay, they're carrying the same or similar genetic information at the same gene loci. Okay, but when you're producing gametes, sperm or egg, and the testes or ovaries respectively, okay, you're only going to pass on one of those two chromosomes. So if you happen to be heterozygous dominant, okay, for a particular trait at the same gene loci on a homologous pair of chromosomes, you're not passing on both. You're only going to pass on one. So therefore, you're either going to pass on to your offspring a dominant allele or a recessive because they're segregated here. Okay? Okay. So we're going to stop right here. Are there any questions? I hope you all have ibuprofen or Tylenol with you, but are there any questions? I know it can get brutal. Let me stop the recording here.